Welcome to this next session of Reproductive. Now, in this session, I have selected some of the class 12 topics, uh, which are important for Neet 2022. Now, the first topic I have selected from reproduction, and this topic is about the gametogenesis, as we have two types of gametogenesis: spermatogenesis and oogenesis. First of all, let us revise spermatogenesis. So initially you have to remember about the stage of spermatogenesis diagrammatically. So spermatogenesis is nothing but male gamete formation or sperm formation. Sperm formation is what we call as spermatogenesis. And you know that the site of spermatogenesis is seminiferous tubules. It is seminiferous tubule, seminiferous tubule, which are some coiled tubules in the testes. And remember from your NCRT that there are 1 to 3 seminiferous tubules in each lobule. 1 to 3 seminiferous tubules in each lobule. Now this is cell from which spermatogenesis occurs. This is the spermatogonium. This is the spermatogonium. This is the cell from where this spermatogenesis starts. You should remember that spermatogonium is a diploid cell, and in this diploid cell each chromosome has a single chromatid. So if you are asked about calculating the total number of chromatids, the total chromatids in each in this spermatogonium will be, as we have 46 chromosomes, that will be 46 into 1, that is 46. Now actually chromosome is having a single chromatid when the DNA has not duplicated, but when DNA has duplicated, chromosome tends to have two separate chromatids. Now this spermatogonium undergoes growth phase. It undergoes growth phase, and in this growth phase, the spermatogonium doubles its DNA. It doubles its DNA. It doubles its metabolites. And it doubles its organelles. And in this growth phase, after this growth phase, there is we get this large cell. This large cell is called as primary spermatocyte. This large cell is called primary spermatocyte. Now you should also note this primary spermatocyte is also a diploid cell, but as a DNA has duplicated, it is each chromosome will be having two chromatids. So if we calculate total number of chromatids, total chromatids will be 46 into, as we have 46 chromosomes into, that is 92 chromatids. Okay, so we will be having 92 chromatids. This is very important number of chromatids. Now this primary spermatocyte undergoes meiosis first. And as a result of meiosis first, we get these cells which are called as secondary spermatocyte. We get these cells called secondary spermatocytes. These are the cells we get after meiosis first. Now remember these secondary spermatocytes, as meiosis first is a reductional division, so we will be having n number of chromosomes in each secondary spermatocyte. So n means 46 by 2, that is 23. So if we calculate total chromatids, that will be equal to n is 23, that is 46 by 2 into, but each you should remember that in meiosis 1 there is no reduction in the number of chromatids, although there is a reduction in the number of chromosomes. So meiosis 1 there is reduction in chromosomes, but there is no reduction in the chromatids. Each chromosome still is in this shape, that is having two chromatids like we have this there in the primary spermatocyte. So we will be having 23 chromosomes and each will be having two chromatids. That means we have total 46 chromatids in each secondary spermatocyte. After this secondary spermatocyte, we have this meiosis second. And in this meiosis second, it is like a mitosis. There is only reduction in the number of chromatids. So the cells will be again n. It is like mitosis, but the chromatids reduce, and we get these cells which are called as spermatids. And these spermatids, they have reduced chromatids. So the total number of chromatids, if we calculate total number of chromatids, that will be equal to 23 into 1. As chromatids have reduced, so it will be equal to 23. So we will be having 23 chromatids in each of these spermatids. Those chromatid calculations very important. You should remember this, via how to calculate chromatid at each stage. So what happens after that, these spermatids get attached to Sertoli cells. And they undergo maturation. And after maturation, they form the tadpole like sperms. they form tadpole like sperms. This is very important. They form tadpole like sperm. And if I talk about an individual sperm, it's individual sperm that has a definite structure. Roughly I will draw that structure, and you should notice all the parts, necessary parts in that structure, so the important parts in the structure include this first part, what we call as head. Second part called as neck, and the third part called as tail. This head contains nucleus. It contains this nucleus, enlarged nucleus, and a short cap on this nucleus, which is called as acrosome. It's a bag of enzymes. So this is called as acrosome. Acrosome is a bag of enzymes which contains those enzymes: zuna, lysine, hyaluronidase, etc. And there are two centrioles in the neck: proximal and distal centriole. These are two centrioles: proximal and distal centrioles. This is proximal centriole. This is proximal centriole. And this one is here distal centriole. Now this black colored thing is the axial filament. This is axial filament, 9+2 flagellate structure. This axial filament. Now notice in this part, axial filament will be covered by this mitochondria. These are mitochondria, and these mitochondria are covering the axial filament. This part is known as middle piece. So this part of the tail in which axial filament is covered by this mitochondria, these are mitochondria here, and this part is called as middle piece. We call this part as middle piece. The next part is called as principal piece, and the last part is called as end piece. Now, that much detail is not required. You just have to remember there is head, there is neck, and there is this tail. These are three main parts, and you remember there is nucleus in the head and this cap called acrosome. After that, remember that there are two centrioles, proximal and distal centrioles, and remember that there is a mitochondrial spiral around axial filament in this part, what we call as middle piece. Okay, now let's go into the second gametogenesis, that is oogenesis. So oogenesis is like spermatogenesis. It is formation of female gametes or ova. So it is formation of ova. Ova formation. This oogenesis is a very complicated process. It starts from these cells, what we call as oogonia. Oogonia are again diploid cells, and the total number of chromatids will be 46 into 2, that is 46 into 1, sorry, that will be 46 into 1. Each chromosome will be having a single chromatid, that is 46 chromatids. Now again, this undergoes growth phase. It forms the largest cell after duplication of DNA, organelles, etc. That cell is called as primary oocyte. Now this primary oocyte, this is our primary oocyte, this is our oogonia. This primary oocyte, if we write down the total number of chromatids, that will be 46 into 2, that is 92. So that is important. Number of chromatids is 92. Number of chromatids is 46. Now again, it undergoes meiosis first, but that meiosis first gets arrested. So it enters meiosis first during the embryonic life. It is important that it enters the meiosis first during the embryonic life. This is very important. This oocyte enters meiosis first in the embryonic life, and in that embryonic life it gets arrested in this meiosis. Meiosis 1. Now it is asked in which phase it is arrested. So it is arrested, remember, it is arrested in diplotin of prophase first. This is a very important question that you might be asked. Diplotin of prophase first is the phase where this is arrested. This primary oocyte is arrested. Remember this. Now this arrested meiosis is released in what we call as during the tertiary stage of folliculogenesis, at puberty, during the tertiary stage of what we call as tertiary stage of folliculogenesis. So at tertiary stage of folliculogenesis at puberty, it is released. This tertiary stage is very important. At tertiary stage it is released, and it forms these two cells. One of the cells is called as first polar body, and second cell is called as secondary oocyte. That is called secondary oocyte. Very important. Second cell is called secondary oocyte. These are both haploid cells, and if we count the number of chromatids, that will be equal to 23 into 2. So number of chromatids will be 23 into 2. Number of chromatids will be 23 into 2, that is 46. That is 46. Very important. Now, this first polar body may undergo meiosis second to form two first polar bodies, to divide two, form two another polar bodies. But these polar bodies are cytoplasmically very deficient cells. These are cytoplasmically very deficient cells. Cytoplasmically deficient cells. These are cytoplasmically deficient cells, and they die. They die. They undergo degeneration. They undergo degeneration or they die simply. Now the secondary oocyte, it enters in meiosis second, but it's again arrested in meiosis second. And which phase you are asked? It is arrested in metaphase second. Metaphase second. It is arrested in metaphase second. And at the time of fertilization, at the time of fertilization, at the time of fertilization, this meiosis second is also completed, and it results in formation of two haploid cells, on one of which is called as second polar body, one of which is called as second polar body, and other is called as definitive ovum. Other is called as definitive ovum, which gets fertilized instantaneously to form the zygote, which gets fertilized to form zygote, to form zygote, which gets fertilized then instantaneously to form zygote. Now again, as the number of chromatids will be reduced, so number of chromatids will be 23 into 1, that is 23, same as that of spermatogenesis. So this is the number of chromatids. You should look at this. So what did we say? It started with oogonia. It started with oogonia, and oogonia undergone growth phase. They formed primary oocytes. Primary oocytes were arrested in meiosis first, in diplotin phase of meiosis first, and then at tertiary stage of folliculogenesis, the meiosis got released, and we got first polar body and secondary oocyte. First polar body degenerated, and secondary oocyte entered meiosis second, but got again arrested in metaphase second. And at the time of fertilization, this meiosis second completed, and we had second polar body and this ovum which gets fertilized into zygote. Now remember one thing, that total polar body, it has been an asked question, total polar bodies, how many total polar bodies are formed. And remember, the total number of polar bodies is 3, as we have two polar bodies right here, which are formed by division of the first polar body and second polar body as a result of meiosis second. So remember this is an asked question, total polar bodies, the answer has been 3. Now, next topic that we revise is important events in embryogenesis. There are several important events in embryogenesis that your NCERT has mentioned, so you should focus on these events because you can get a question from this. First important event is formation of heart, and this heart is formed at the end of first month. So at the end of first month, you have the heart formed in the embryo. Now formation of limbs and digits, it occurs at the end of second month. So in second month, the limbs and digits of the fetus are formed. Then major organ systems are completed in third month. So third month marks the completion of the major organ system formation in the fetus. Then in the fifth month, you observe hair on the head of the fetus, and you also observe the movements. So movements of the fetus and hair on the head are observed after the fifth month. This is very important. And then you have these separate eyelids. This is very important key term, separate eyelids and eyelashes along with fine body hair. This is completed at the end of sixth month. So at the end of 6 months, you have the completed fetus formation. Almost everything is formed in the fetus except the lungs, except the maturation of the lungs. So importantly, remember, formation of heart occurs at the end of first month occurs in first month. Formation of limbs and digits occurs in the second month. Formation of major organ systems, it occurs in third month. Formation of movement and the hair on head is formed at the end of fifth month. Separate eyelids at the end of sixth month. Very important. So these were some important events of what we call as embryogenesis. You can remember a hint, we have one heart, so our heart is formed in first month, 1 month, or first month. And we have two limbs, and those limbs are formed at the end of second month. This hint you can use here if you want to remember these two tricks, two major organs importantly. Then next topic I have selected from reproductive health, that is intrauterine devices. You know that intrauterine devices are the most widely accepted contraceptive methods in India. This is the most widely accepted contraceptive method. This is important, has been written in our NCERT. Very important, written in our NCERT. And these are the devices which are fixed in uterus by a gynecologist. They are fixed in uterus by a gynecologist, by a specialist who is always a gynecologist. Now there are three types of IUDs. You have these non-medicated IUDs, you have these hormone releasing IUDs, and you have these copper releasing IUDs. So some of these devices do not contain any medicine. They are called non-medicated IUDs, and we have this example of Lippes loop. This Lippes loop, what does it do? It increases phagocytosis. It increases phagocytosis inside the uterus. So it increases the rate of phagocytosis inside the uterus so that the sperms are not able to reach the fertilization site of fertilization. Now we have these hormone releasing IUDs. These hormone releasing IUDs release some hormones, mainly progesterone, and we have two examples, progestasert and LNG20. Very important. Progestasert and LNG20 are two examples of hormone releasing IUDs which release hormones inside the uterus. And these, what do? They decrease the chances of ovulation. They decrease the chances of ovulation, and also they decrease the chances of implantation. So they make uterus unfavorable for implantation, and they also increase the cervical mucus consistency, cervical viscosity. They also increase the cervical viscosity. These all three things have been written in your NCRT, so you better focus on them, remember them. Most importantly, they increase the cervical viscosity. Then we have these copper releasing IUDs which release copper. And you know there are various devices, copper T, copper 7, and multi-load 375. What does copper do? Copper 2 positive ions suppress the motility of sperms. Suppress motility of sperms. So motility of sperms is inhibited by copper ions, and that is the mechanism of action of copper releasing IUDs. You should always remember them. Then I have selected these assisted reproductive technologies. Assisted reproductive technologies. Again, from the reproductive health, very important. We have four technologies. IVF, which is called as in vitro fertilization. In vitro fertilization. In vitro fertilization. What do we do in IVF? We collect the gametes. We collect sperms and ova. We collect sperms and ova. We collect sperms and ova. The sperms and ova are collected. They are fused. They are fused in lab, in a stable dish. That's why we also call it test tube baby program. We also call it test tube baby program. This is also called as test tube baby program. It's also called as test tube baby program. So what we have collected the sperms and we have fused them in lab, and we get these embryos. Then what we do, if the fallopian tube of the female is normal, then we transfer the embryo. With less than 8 blastomers or cells. Into the fallopian tubes. And that is called as zygote intrafallopian transfer. That technique is called as zygote intrafallopian transfer. But what if the fallopian tube is not normal? What if fallopian tube is defective? If the fallopian tube is defective, then we transfer the embryo with more than 8 blastomers. We transfer this embryo with more than 8 blastomers into uterus directly. And that is called as IUT, that is intrauterine transfer. That's called as intrauterine transfer. So in vitro fertilization, what we have done, we have collected the sperms and ova, we have fused them in lab, we have got the embryos, and if fallopian tube is normal, we transfer the embryo with less than 8 blastomers into fallopian tubes by the technology called as ZIFT. And if fallopian tube is defective, we transfer the embryo with more than 8 blastomers into the uterus. That's called intrauterine transfer. And we know this IVF as test tube baby program. Now we have second one, that is called gamete intrafallopian transfer. What we do in gamete intrafallopian transfer, we collect the ova and deliver it into fallopian tubes. Their fertilization occurs normally, so it is an in vivo technique. Fertilization occurs in the fallopian tube normally as it happens. So what is the full form? Gamete intra fallopian transfer. Gamete intra fallopian transfer. Then we have artificial insemination, which is quite explanatory. Artificial insemination, where we put the sperms into the uterus or vaginal canal. So what happens? Manual delivery. We have to manually deliver the sperms into uterus or vagina. Into uterus or vagina. That is called as artificial insemination. Then we have this ICSI. What do we mean by ICSI? ICSI means intracytoplasmic sperm injection. Intracytoplasmic sperm injection. Intracytoplasmic sperm injection. What do we do in this intracytoplasmic sperm injection? We inject the sperms directly into the what we call as ovum, cytoplasm of ovum, where fertilization occurs. It's especially used when there are immotile sperms. It's especially used when the sperms are immotile. So intracytoplasmic sperm injection, the sperm is directly injected into the ooplasm, and it is especially used when the sperms are immotile. Now next topic I have selected from genetics, that is about the 7 contrasting traits used by Mandel. Sometimes it is asked, it is written in your NCRT. So you remember it like this: there are 3 colors, there are 2 shapes, and there is position and height. 3 colors, 2 shapes, position and height. So height of the stem is taken as a character. Tall is dominant, dwarf is the recessive. Then color of the flower is taken where you have violet and white, and position of the flower is taken where you have axial and terminal. Shape of the pod is taken where you have inflated and constricted. Color of the pod is taken where you have green and yellow. Shape of the seed is taken where you have round and wrinkled, and color of the seed is taken where you have yellow and green. So you should remember all these 3 colors, 2 shapes, position and height. 3 colors, 2 shapes, position and height. So color of flower, color of pod, and color of seed. 2 shapes, shape of pod and shape of seed, and position is of the flower and height is of the stem. Okay, so we have shapes and colors of pods, and we have also color of the flower, and we have position of flower, and we have height of the stem. Then I have selected this types of sex determination, chromosomal sex determination. This is very important topic. You can get one of the questions from this types of chromosomal sex determination. So you have 3 types mainly: XX, XY. XX, XY means it is a male heterogametic type of sex determination, where you have females with 2 X chromosomes and males with X chromosome and Y chromosome. So male has 2 different chromosomes, X and Y. So male is heterogametic, which is very important, and female is homogametic, as both the gametes will be containing X chromosomes. And this is present, this type of sex determination is present in humans and Drosophila. It is present in humans and Drosophila. It is also called as male heterogamy. Then you have XXX0, where female is having 2 X chromosomes, male is having only 1 X chromosome, and not any type of Y chromosome, not any other type of chromosome. So male is again heterogametic. Male is again heterogametic, as male has some of the sperms will get X chromosome, some of them will not. So it's also female male heterogamy, and it has been observed in grasshoppers. It has been observed in grasshopper. And then we have ZWZZ, where females are having Z chromosome and W chromosome, and males are having 2 identical Z chromosomes. So females are heterogametic. Females are heterogametic. So it is female heterogamy. It is female heterogamy type of sex determination. And where do you observe it? You observe this in birds. Very important. You should remember this. You observe this in birds. You observe this in birds. So you observe this in birds, and birds have ZWZZ type of sex determination, where females have Z chromosome and W chromosome, and males have 2 Z chromosomes. Okay. Then next I have taken these symbols used in pedigree analysis. Very important, these can be asked. You have the males, which are represented by square, females which are represented by circle, and unspecified gender, which is represented by this what we call as rhombus like figure, pyramid like figure. And whenever the individuals are affected, you shade them with red color or any other color. And whenever you have to show the mating, you draw a line between the male and female. Whenever you have to show the consanguineous mating or the mating between close relatives, you show this by a double line. This has been asked. And children are shown by this vertical lines, and from birth order, left to right is the birth order, left to right is the birth order. It means that first child is female, second child is male, and then affected male child will be shown like this. And if we have to show multiple male childs affected or unspecified genders affected, we can simply put a numeral in the center of the shape. Then I have selected these three trisomies which you observe in the chromosomal anomalies. These three trisomies which are important, out of which there is Down syndrome, which is trisomy of the 21st chromosome, which is trisomy of the 21st chromosome, which is trisomy of 21st chromosome. It's trisomy of 21st chromosome. Remember, the total chromosomes will be 46, 47, and autosomes will be autosomes will be 45, and sex chromosomes will be normal. So it can be 45 plus XX, or it can be 45 plus XY. So both males and females can be affected, and the total number of chromosomes will be 47, and 45 plus XX, 45 plus XY. There is an extra 21st chromosome. That means there are three 21st chromosomes. There are three 21st chromosomes. Three 21st chromosomes. This is a trisomy of 21st chromosome. Then we have Klinefelter syndrome, which is X chromosome trisomy in males. So what you have total number of chromosomes 47. Total chromosomes is 47, and this 27 is equal to 45 plus X. 40. Sorry, it's equal to 44 plus. It's equal to 44 plus XXY. So there's an extra X chromosome in case of male. There's an extra X chromosome in case of male. So these are usually infertile males. These are infertile males because of this extra X chromosome. Then we have Turner syndrome, which is monosomy of X chromosome. Turner syndrome, which is monosomy of X chromosome. And what is the total number of chromosomes? Total number of chromosomes will be 45. And these 45 chromosomes will be having 44 sex chromosomes and X0 sex chromosomes. So this is a female, as there is no Y chromosome, but there is not an extra chromosome either. So there is one X chromosome is missing, and there is 0 instead of it. 0 means it is not there. This X chromosome is missing. So total chromosomes are 45, out of which autosomes are normal 44, and 6 chromosomes are X0. One is X and other one is not there. Okay, now we'll study some of the important aspects of Human Genome Project. Very important. So salient features Human Genome Project. What Human Genome Project has revealed, what information we have got from the Human Genome Project. So first information is about the number of base pairs. So we have found that there are 3164.7 million base pairs, which is approximately equal to 3 billion base pairs, which is approximately equal to 3 billion base pairs. Now second thing that we have got from this is the average number of genes is average gene contains about 3000 bases, but there is a large amount of variation. The largest gene dystrophin is having 2.4 million bases. So largest gene dystrophin is having 2.4 million bases. Now another thing that we have got is that total number of genes is about 30,000, which is lower than the estimate which was made 80,000 to 1,40,000. And 99% nucleotide bases are exactly same in all the people. This is an important deduction that we have made from this thing. This important deduction we have made from this thing. 99% nucleotide bases are exactly same. So total number of genes is 30,000. Average gene contains 3,000 bases, and we have 3164.7 million base pairs, or 3 billion base pairs. Now another important thing, less than 2% genome codes for proteins. So protein coding genome is less than 2%. This is very important. And we have a lot of DNA which is made up of repeating sequences. This is very important. This forms the basis of DNA fingerprinting, which we will read subsequently. Then it is describing about these repetitive sequences. These are stretches of DNA that are repeated many times, sometimes 100 to 1,000 times. And they shed light on chromosome structure, dynamics, and evolution. Now important thing about the structure of chromosome, the first chromosome is the most is having most number of genes. The largest chromosome is having 2968 genes. First chromosome is having 2968 genes, while the Y is having the lowest amount of genes, about 231. Chromosome Y is the smallest chromosome, while as chromosome 1 is the largest chromosome. And most important, scientists have identified about 1.4 million locations where single base DNA differences occur, which are called as SNPs. SNPs, yeah, synapse. single nucleotide polymorphisms. And this information promises to revolutionize the process of finding chromosomal locations for disease associated sequences and tracing human history. Okay, so important things: we have how many base pairs? We have 3 billion base pairs. We have 3000 base in every gene, but dystrophin is having 2.4 million. It is largest gene on X chromosome. Total genes is 30,000, and 50% genes have unknown function. 99% bases are same. Less than 2% codes for proteins. Repeated sequence form a large portion of DNA. One is largest with 2968 genes. Y has fewest, and 1.4 million locations have SNPs, single nucleotide polymorphisms. Okay, that is some information from the Human Genome Project. Thank you for this session. This precise session was like this, and we will choose the rest of the syllabus in next session. That's all for today. Have a nice